Hello everyone, I am Dr.VRAO. From the lecture 72 of Catalysis Part 1, we came to know that in homogeneous catalysis, rate of reaction is directly proportional to concentration of catalyst, whereas in heterogeneous catalysis, rate of reaction is directly proportional to surface area of the catalyst, right. Moreover, life is possible due to the coordination of various reactions that takes place in living organisms. For example, digestion of food, absorption of appropriate molecules and ultimately producing energy. These processes involves a sequence of steps under mild conditions with the help of biocatalysts called enzymes. Enzymes are globular proteins having molar mass of order 10,000. Such processes are, for example, in our digestion, first of all, starch converted into maltose by enzyme diastase, and then maltose converted into glucose, which is being catalyzed by maltase. These are the sequence of reactions that takes place in our body biology, right. First, starch converted into maltose, which is being catalyzed by diastase, enzyme diastase, and then maltose is converted into glucose, which is being catalyzed by enzyme maltase, right. These are the sequence of steps, right. And one more thing is that these enzyme catalytic reactions are needed very small quantities of enzymes, I repeat, it is the very important point, right. These enzyme catalytic reactions needed very small quantities of enzymes for the progress of reactions, right. And this is the these are the points that we learned in the lecture 72, right. Now in this class we shall study kinetics of enzyme catalyzed reactions, and we know that enzyme catalyzed reactions comes under the category of homogeneous catalysis, right. So for study of this enzyme catalyzed reactions, Michaelis Menten proposed a mechanism, right. Michaelis Menten proposed a mechanism, right. In this mechanism they proposed that first enzyme reacts with substrate is nothing but reactant, okay. Enzyme reacts with substrate, forms enzyme substrate complex, and this formed enzyme substrate complex is in equilibrium with the enzyme and substrate, right. This is forward reaction rate constant and this is backward reaction constant K inverse. Usually it happens very fast, it is a fast step, and then enzyme substrate complex decomposes to give products plus again enzyme, right. This is step 2 and this step takes place very slowly, right. So they proposed this mechanism, right. First enzyme reacts with substrate, form enzyme substrate complex, and this enzyme substrate complex is in equilibrium with the enzyme and substrate, right. And then this takes place very fast, and then enzyme substrate complex decomposed to give products plus again enzyme. This is very slow step. So, usually the rate of this entire process depends on the slow step. We all know rate is determined by slow step of the process, right. So, rate of this process, rate is equal to K2 into concentration of ES, concentration of enzyme substrate complex, right. So, rate is directly proportional to concentration of enzyme substrate complex, and rate is equal to some constant K2 into enzyme substrate complex, right. Already I told you that enzyme catalyzed reactions are needed very small quantities of enzymes for the progress of reactions. Okay. So, this enzyme catalyzed reaction comes under the category of homogeneous catalysis. In homogeneous catalysis, rate is directly proportional to the concentration of catalyst. Okay. You just remind that statement. Okay. So, rate is equal to K2 into concentration of enzyme substrate complex, concentration of enzyme substrate complex. Okay. Then according to previous statement, that is enzyme catalytic reactions needed very small quantities of enzymes for the progress of reaction. That means enzymes are needed, that the concentration of enzyme is needed very less than concentration of substrate. That means when compared to substrate, enzyme concentration is very low. Okay. So, when enzyme concentration is less than concentration of substrate, consequently the formed enzyme substrate complex is also less than substrate concentration, because enzyme concentration is very low. So, that enzyme forms complex with substrate. So, that enzyme substrate complex is also in low concentration, right. So, according to known statement, enzyme substrate concentration is enzyme substrate complex is very low, but for this reaction rate is given by rate is equal to Kt into concentration of enzyme substrate complex. But we know that enzyme substrate complex is in very low concentration. How can we measure? But we need to substitute concentration of enzyme substrate complex to get the rate of this reaction. But enzyme substrate complex is very low. This concentration is very low. How can we measure that? How can we measure such low enzyme substrate complex concentration? Right? How can we measure such low concentration of enzyme substrate complex? Right? We can measure with the steady state approximation method. Okay? That is, steady state approximation enable us to measure the such low concentrations. Okay? That is the advantage of steady state approximation. Okay, steady state approximation enable us to measure such low concentrations, right. So, with the help of steady state approximation, we can measure such low concentration. So, let us write steady state approximation. Actually, this steady state approximation needs another 2 or 3 classes. For the sake of my convenience, I write the steady state approximation equation directly. Okay. So, d by dt of concentration of ES is given according to steady state complex. In this process, enzyme is consumed in the forward reaction and enzyme substrate complex is consumed in the backward reaction, and again enzyme substrate complex is consumed in the second step. Okay. So, we can write K1 into concentration of E into concentration of S minus K inverse into concentration of ES minus K2 into concentration of ES is equal to 0, right. Okay. So, in order to get the concentration of ES, concentration of enzyme substrate complex, we need to substitute enzyme concentration. Okay, enzyme concentration, that is concentration of E. But concentration of E, that is free enzyme concentration, that free enzyme concentration cannot be measured experimentally. Then how can we measure free enzyme concentration? We can measure free enzyme concentration with the enzyme conservation equation. Enzyme conservation equation. Enzyme conservation equation is given by total enzyme concentration is equal to free enzyme concentration plus enzyme substrate complex concentration, right? So, concentration of E naught means it is total enzyme concentration, and concentration of E means free enzyme concentration, and concentration of ES is enzyme substrate complex. From this equation we can measure free enzyme concentration. From this we can measure free enzyme concentration is equal to concentration of free enzyme is equal to concentration of total enzyme concentration minus enzyme substrate complex, right? Okay. So, we can get free enzyme concentration. When we substitute this free enzyme concentration in this equation, we get enzyme substrate complex concentration, right? From this equation we get free enzyme concentration. When this equation is substituted in this equation, we get enzyme substrate complex concentration. When we get enzyme substrate complex concentration, we can substitute in this equation, we get rate. So, when we solve these processes and when we substitute this equation in this equation and we get the concentration of enzyme substrate complex, and then it is substituted in this equation and simplifying, we get a final equation. I do not go through all the derivation because due to time bound limits, right? When all these processes done, we get a final equation, that is rate is equal to K2 into concentration of E naught into concentration of S by Km in plus concentration of S. This is called Michaelis-Menten's equation. This is Michaelis-Menten's equation. Michaelis-Menten's equation, right? Equation, and this is called Michaelis-Menten's constant. Km is equal to K inverse plus K2 by K1. This is the Michaelis-Menten's constant. Michaelis-Menten's constant, right? So, this is the important equation, right? Now, simplifying this equation. Now, let us simplify this equation, right? Let us simplify this equation. Okay. We know that this enzyme catalytic reaction comes under the category of homogeneous catalysis. In homogeneous catalysis, rate is directly proportional to the concentration of catalyst. Okay. For example, if when all the enzyme concentration is over, when all the enzyme concentration is exhausted, that means all the enzyme concentration is exhausted, that means rate of reaction is maximum, because in homogeneous catalysis rate is directly proportional to concentration of catalyst. When catalyst is all the concentration of catalyst is exhausted, that means rate is maximum, right? Okay, simple, you don't there is no ambiguity, there is no difficulty. In homogeneous catalysis, rate is directly proportional to the concentration of catalyst. Okay, enzyme catalytic reaction comes under the category of homogeneous catalysis. Okay, so when enzyme concentration is, when all the enzyme concentration is over, when all the enzyme concentration is over, when all the enzyme concentration is over, okay, that means rate is maximum, rate is maximum. Okay, so when all the enzyme concentration is over, when all the enzyme concentration is over, there is no free enzyme concentration. Okay, when all the enzyme concentration is exhausted, there is no free enzyme is remained. Okay, then this equation becomes E naught equal to Es. Okay, that is total enzyme concentration is equal to Es, enzyme substrate complex, enzyme substrate complex. When all the enzyme is exhausted, rate is maximum, rate is maximum, that is this rate becomes maximum, or maximum or velocity maximum, rate maximum or velocity maximum is equal to K2 into concentration of Es. And now we can replace this concentration of enzyme substrate complex with K2 into E naught, K2 into E naught, because when all the enzyme concentration is exacted, enzyme substrate complex is equal to total enzyme concentration, K2 into E naught. Okay, so rate is maximum or velocity is maximum, then K2 equal to E naught. Okay, so we can write K2 into E concentration of E naught. We instead of K2 into E naught, we can write V maximum. So therefore, R maximum, so rate maximum is equal to K2 into E naught. This can be written as K2 in V max. This can be written as V max into concentration of S by Km plus concentration of S. This is called simplified Michaelis-Menten's equation. This is called simplified Michaelis-Menten's equation, right? Simplified Michaelis-Menten's equation. This is very, very important. From this we can discuss three conditions. From this we can discuss three conditions. Okay, let us discuss the three conditions. Okay, so rate is equal to, this is simplified Michaelis-Menten's equation, equation. Okay, from this we can discuss conditions. Condition 1. Okay, condition 1. When Km Michaelis-Menten's concentration, when Km is greater than substrate concentration, when Km is greater than substrate concentration, we can, Km is greater than substrate concentration, so we can neglect the substrate concentration. Okay, we can neglect the substrate concentration, therefore rate is equal to V max into concentration of S by Km. Okay, we can write these two constants as K dash into concentration of S. So rate is equal to K dash into concentration of S. From this rate equation, we can interpret that this is a first order rate equation. This is first order rate equation. Okay, when Km is greater than concentration of substrate, then Michaelis-Menten's mechanism, according to Michaelis-Menten's mechanism, that enzyme catalytic reaction follows first order mechanics, right? Then case 2, when concentration of S is greater than Km, concentration of substrate is greater than Km, then in this denominator we can neglect the Michaelis Menten's constant. Therefore, rate equal to V max into concentration of S by concentration of S. Okay, and this S is cancelled. Therefore, V max rate is equal to V maximum, that is velocity maximum. From this we can interpret that that follows zero order. This is a zero order rate equation. This is a zero order equation. Okay, and case 3. Case 3. When substrate concentration is equal to Km, therefore rate equal to V max into concentration of S, where instead of Km we can write concentration of S because these two are equal. Okay, therefore V max into concentration of S by 2 into concentration of S. This SS get cancelled. R is equal to V maximum by 2. V maximum by 2. When substrate concentration is equal to Michaelis Menten's constant, rate equal to velocity maximum by 2, half maximum, half velocity maximum. Okay, let us represent all these three conditions in a graph, right? In a graph. I shall draw the graph. Okay, so all these three conditions are represented in this graph. Okay, graph is plotted between rate and concentration of substrate, because from this is y is equal to, this is you can imagine that y is equal to mx, y is equal to mx, that is y. When we plot between y and x, that is rate and substrate concentration that pass through the that is a straight line pass through the origin. Okay, and it follows first order and then zero order. Okay, so when we observe this graph, with increasing the substrate concentration, reaction kinetics changes from first order to zero order, and when concentration of rate, Michaelis Menten's constant is equal to concentration of S, that is V max by 2. Okay, from this graph we can observe that with increasing the substrate concentration, kinetics changes from first order to zero order. Okay, so usually questions are being asked from this area. Okay, asking these three conditions and explaining this graph, right? There is a certain reason. For why this kinetics changes from first order to zero order. Okay, zero order. There is a reason for changing from first order kinetics to zero order kinetics. Okay, why that enzyme catalytic reactions changes from first order kinetics to zero order kinetics? Why? Because enzyme catalytic reactions, in this enzyme is participating. Enzyme has one or more active sites. Where substrates are attached in order to catalytic reaction to takes place. Substrates are attached to active sites of the enzyme. Okay, that means enzyme has one or more active sites where substrates are being attached in order to enzyme catalytic reaction to takes place. Right. When substrate concentration is low, the number of active sites, okay, or the enzyme active sites are unoccupied. Okay, I repeat, when substrate concentration is low, then the number of active sites of enzyme are unoccupied. unoccupied because substrate concentration is very low. When substrate concentration is low, the number of active sites of the enzyme are unoccupied. When substrate concentration goes on increasing, the number of active sites of enzyme are occupied by substrate molecules. The number of active sites of enzymes are occupied by substrate molecules. Okay, right? When substrate concentration is moderate concentration, that means substrate molecules are attached to the active sites of the enzyme. Okay, that means active sites of the enzyme are that means substrate molecules are being attached to the active sites of the enzyme. But when substrate concentration is very high, that means very high, that means substrate concentration is very high, there are no active sites vacant to attach the substrate molecules. That means there are no active sites are unoccupied because all the active sites of the enzymes are occupied by substrate molecules. So, because there is no unoccupied active sites at the enzyme, therefore reaction kinetics follows zero order. When enzyme catalytic reaction follows zero order, that means there are no active sites at enzyme. That is, there are no active sites are available at enzyme, or all the enzyme active sites are occupied. Okay, at moderate concentrations, some of the active sites of the enzyme are unoccupied. So, it follows first order kinetics. But high concentration of substrate, there is no vacant active sites at enzyme, it follows zero order. Okay. So, this is about Michaelis Menten's equation. Okay. So, they are asking case 1, case 2, case 3, and based on this plot questions are being asked. Okay, I hope you this lecture is helpful to students, right? Thank you for watching.